about, they are not so easily passed through. So the correct answer is first potassium, then comes sodium, then depending on the charge, amount of charge which is there, magnesium as well as calcium. Now, in the biological membrane, the integral proteins and the lipids interacts mainly by, as we know that lipids are the hydrophobic nature, and these integral proteins which are there, they will be having a kind of amino acids present in between the membrane or the hydrophobic region of the membrane, so that the interaction between these two will be hydrophobic plus hydrophobic. So generally it will be hydrophobic interactions. Now, another technique which we usually perform for the assessment of the transport of membranes across the transport of molecules across the membrane when we are talking about, that is the FRAP technique, that is the fluorescence recovery after photobleaching. That is how the kinetics or the rate of diffusion in the living cells is done, or moving these objects with the help of a fluorophore and using a fluorescence microscopy. Now, the only thing what we have to see is the equipment must be able to bleach a defined area of the membrane so that this will be easy for analyze. The order of the predicted rate of diffusion across the lipid bilayer, when we are talking about highest to the lowest, when we talk about, is depending on the polarity of the compounds or the ions which we come across the substances. Then when we talk about these ions or the molecules or the solvents, when we are talking about, from the highest to the lowest, when we are talking about, it will be highest non-polar to the lowest non-polar, or we can say that from non-polar to the polar. So the sequence will be toluene, phenylalanine, galactose and chlorides. Now the next question, when we talk about fibroblast cells, you might have come across, and normally all the cells, when we consider, including the neurons which are there, they are negative, having a negative electric potential, as we have mentioned earlier, because of the high amount of proteins which are present inside the cells. Then the change in the membrane potential is done mainly by the two ions, the sodium and potassium, by the movement of these ions inside and outside. Now, when we talk about the fibroblasts, these ions also can be actively moved in or outside. But when we compare with the neurons, these fibroblast cells, they do not generate an action potential. Now, this action potential in the neuron or the neuron cells when we are talking about, or the nerve cells when we are talking about, you know that the action potential will be created only after achieving a threshold potential. And once it achieves the threshold potential, so till that time, the passage of the sodium into the cell will be with respect to the normal diffusion process or facilitated diffusion of the sodium. Then comes sudden opening of the voltage gated channels, so that all the sodium or maximum number of sodium will be entering into the cell. Whereas when we are talking about the fibroblast cells, most of the time, or we can see that there is the absence of these voltage gated ion channels, so the amount of sodium entering into the fibroblast cells will be less, therefore the action potential, or it may not be achieving the threshold potential, therefore the action potential cannot be created. With this, we will finish the basic aspects of the membrane transport mechanism. With this, we will be stopping the lecture or on the membrane transport mechanisms or the basic membrane structure and function. And I would like to thank again the GSPTM officials as well as the BISAC people who helped in recording this lecture. Thank you very much.
Vande Gujarat.